My name is Valeria Chichigova, and I'm an Associate Director of IPSC Technology and Head of Retina at Newcells Biotech. In this presentation, I will be talking about retinal organoids being used as a tool for in vitro preclinical testing. There are a number of limitations of current preclinical retinal models. Animal models do not recapitulate the physiology of the human retina. Human retinal explants have a restricted window of use, and tissue access is challenging. Primary cells and cell lines of single retinal cell types do not mimic the interaction of multiple retinal cell types. So we've developed a human iPSC-derived 3D retinal organoid model, which we're now manufacturing regularly and making it accessible for wider use. At day 150 of the differentiation, retinal organoids contain major retinal cell types, including photoreceptors, retinal ganglion cells, horizontal amacrine and bipolar cells, and mural glia. The organoids are approximately 1.3 millimeters in diameter. We grow them in 96 plates for increased throughput. And organoids are manufactured on a rolling program every 4 weeks. At day 150 of the differentiations, they're QC'd. We've performed a number of different tests and showed that the organoids are stable at room temperature, so they're shipped as a ready-to-use assay globally. Retinal organoid generation follows the developmental timeline of retinogenesis in vivo. So depending on the cell type of interest, the organoids can be used at different stages. So for instance, at day 60 of the differentiations, retinal ganglion cells are more abundant, whereas for bipolar cone and rod photoreceptors are more prevalent at day 180. At day 150 of the differentiations, the organoids contain major retinal cell types, which are organized in a laminated manner. The organoids can be used for various applications, including disease modeling, gene therapy, drug safety and efficacy screening, and developmental studies. Today, I will be talking about using organoids for gene therapy and drug safety and efficacy screening. So together with our collaborators at Oxford University, we've shown that the organoids can be successfully transduced with AAV vectors. Reported gene expression was observed as early 5 days post-transduction and increased over time. Type promoter enabled transduction across a range of different cell types, whereas GRK1 vectors were expressed in photoreceptors. GRK1 vectors provided a clear ring of GFP expression in the outer layer cells of the retinal organoids, and this is where we would find photoreceptor cells. So we've cross-sectioned the organoids and showed that there was positive colocalization with recoverin stain cells. So these are, the recoverin is a pan-photoreceptor marker. We have also used the known retinotoxins with the organoids and shown that the organoids respond to compounds known to induce retinal toxicity in a dose-response manner. Known non-toxic compounds had no effect on viability. To address the limitation of the retinal organoid model in that it doesn't contain an immune component, we've co-cultured the organoids with microglial cells, which were derived from human iPSCs, and then we've tested them at various time points of the differentiation. So firstly, we found that retinal organoids retain morphological features when co-cultured with microglial cells, and also we've done single-cell RNA-seq analysis and showed that there was still presence of key retinal cell populations in the organoids containing microglial cells. So to conclude, hopefully I was able to demonstrate that the iPSC-derived retinal models are an emerging in vitro tool for various applications. They have a potential of being used in efficacy and safety screens of new compounds and for disease modeling applications. They're increasingly showing utility in gene therapy applications and can be used for testing novel gene therapy vectors and investigating optimal doses, therapeutic potential and safety, and as a tool for studying retinal disease mechanisms and development of treatments. Here at Newcells, we're making the organoids accessible by producing regular batches that can be shipped internationally. I would like to thank my colleagues here at Newcells and our collaborators from University of Oxford, Roche, Merck, Novartis, Newcastle and Northumbria universities for supporting this work. Thank you.